For understanding the concept of gluconeogenesis, we first need to revise the process of glycolysis and Krebs cycle. So starting from glycolysis, it was a 10-step process that generated pyruvate molecule. Now there was glucose that was converted into glucose 6-phosphate. This glucose 6-phosphate converted itself into fructose 6-phosphate. After that it was converted into fructose 1,6-bisphosphate. Now this element was converted into two compounds known as glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Glyceraldehyde 3-phosphate was used further, and it was converted into 1,3-bisphosphoglycerate. This was then converted into 3-phosphoglycerate, then 2-phosphoglycerate, and then it was converted into phosphoenolpyruvate that gave rise to pyruvate molecules. Now gluconeogenesis means this pyruvate will be converted back into glucose, but it will not be following the same pathway of glycolysis. Why is that? Because, as you can see here, there are 3 points where this glycolysis is irreversible. For the process of gluconeogenesis, this reaction must pass these 3 steps, which are irreversible. So what happens is that whenever there is low blood glucose levels, lactic acid from the muscles comes and convert itself into pyruvate molecule. So now pyruvate convert itself into acetyl coenzyme A with the presence of enzyme known as pyruvate dehydrogenase. This process proceeds towards Krebs cycle. But in gluconeogenesis, pyruvate converts itself directly into oxaloacetate in the presence of enzyme known as pyruvate carboxylase. Now this oxaloacetate is converted into malate. All of this is going on the mitochondria. But one unique thing happens. This malate moves outside the mitochondria. After moving outside, it converts itself back to oxaloacetate. Now this oxaloacetate is a 4-carbon compound which must convert itself into phosphoenolpyruvate, which is a 3-carbon compound. For this, it needs to remove its 1 carbon, and there will be release of carbon dioxide. Now with this removal of carbon dioxide will give rise to addition of a phosphate group. This addition gives rise to phosphoenolpyruvate. The reaction is aided by the enzyme known as PepCK or phosphoenolpyruvate carboxykinase. Now the main process of glycolysis will be followed, but it will again get stuck at fructose 6-phosphate. To aid this conversion, an enzyme is involved known as fructose 1,6-bisphosphatase. This enzyme will convert it into fructose 6-phosphate and reaction will proceed again. But it will again get stuck in glucose 6-phosphate. For conversion of glucose 6-phosphate into glucose, there is a special structure in the cell known as smooth endoplasmic reticulum. This smooth ER has transporters that enters glucose 6-phosphate inside it. It contains a special enzyme which will convert this glucose 6-phosphate into glucose molecule, and the enzyme is known as glucose 6-phosphatase. Now this converted glucose will move outside this smooth ER and exit the cell for further utilization. So, this was about the gluconeogenesis through lactic acid. Now let us talk about fats now. Basically, triglyceride is involved in gluconeogenesis. They have a glycerol part and fatty acid side chains, which are 3 in number. Both parts are involved in gluconeogenesis. If we talk about glycerol, it needs to add a phosphate group to convert itself into glycerol 3-phosphate with the presence of the enzyme known as glycerol kinase. This glycerol converts into dihydroxyacetone phosphate. You must remember that this dihydroxyacetone phosphate is interconvertible into glyceraldehyde 3-phosphate, which is an important step of glycolysis. Now the rest of the steps remain same as lactic acid gluconeogenesis. Now let us discuss these 3 fatty acid chains. They are known as odd-chain fatty acids, and they can undergo beta oxidation. This is an insignificant process that converts fatty acids into acetyl coenzyme A. Then the rest of the process goes same as lactic acid. It is mostly a negligible process. So, lastly comes amino acid. Amino acids can undergo the process of transamination. Let us first understand what is it. So basically, when an amino acid reacts with an enzyme, they form alpha ketoside with combination of enzyme along with amino group. Now this combination further reacts with alpha-ketoglutarate and the enzyme gets separated with the formation of glutamate. Now this newly formed keto amino acid can act on various steps of Krebs cycle. This Krebs cycle ultimately leads to formation of moly and undergo the process of gluconeogenesis. This newly formed amino acid can act on pyruvate. It can also act on acetyl coenzyme A. Then it can also go towards alpha-ketoglutarate and on succinyl coenzyme A as well. So this was all about gluconeogenesis and the sources from which glucose is made from non-carbohydrate sources. Let us move towards its regulation now.